Okay, this is an introduction to the photosynthesis lab. And I'm going to spend some time talking about different types of approaches we have for measuring net photosynthesis in trees and other plants. Recall that one of the fundamental things about net photosynthesis is that net photosynthesis takes up CO2 and releases oxygen. So those are the two main ways that we measure the rate of net photosynthesis. We can measure the rate of CO2 uptake. We can measure the rate of oxygen evolution. Both of those are directly related to the rate of net photosynthesis. There are approaches that use oxygen release, but I'm not going to talk about those. They use oxygen electrodes and leaf discs usually. Most measurements of net photosynthesis in the field use rate of CO2 uptake to measure rates of net photosynthesis. So the heart of any photosynthesis measurement that's measuring based on CO2 is an infrared gas analyzer. Sometimes it's abbreviated IRGA, I-R-G-A. And that's basically an infrared gas analyzer. It's just an instrument that can measure the concentration of CO2 gas in an airstream. And this is the basic design of an infrared gas analyzer. So what we have here is an infrared source, and then a hollow tube here, and then an infrared detector. And then we have a port. We can put gas into that tube, and then of course port where the gas can leave the tube. Okay, so the way this works is that infrared source shines infrared radiation through that tube to the detector. The detector can detect how much infrared radiation is reaching this side of the tube. Let's imagine a situation where the gas in here has no carbon dioxide. The important thing to know is carbon dioxide absorbs strongly in infrared wavelengths. So if there's essentially no CO2 in this gas, all of the infrared radiation that leaves the source arrives at the detector. We will be able to detect that as a signal. Now let's say, for instance, that the gas that we're putting through this tube has some amount of CO2 in it. Those CO2 molecules in the gas will absorb infrared radiation in this tube, and not all of the infrared radiation then will make it to the detector because it's been absorbed by CO2 molecules in between. So the strength of the infrared detection at the detector is related to the amount of CO2 in the gas in the tube in between the source and the detector. And by calibrating the signal from the detector, use it by flowing different known rates of CO2 through this tube, we can calibrate this detector, and that can tell us the CO2 concentration in some unknown gas stream. This ability to measure CO2 concentration is the basis of all photosynthesis measurement systems that use CO2. So I'm going to talk about how a photosynthesis measurement system is put together to measure photosynthesis rate. So all gas exchange systems need some type of chamber into which you place a leaf to measure its photosynthesis rate. So the schematic I have here is just this square, and we have a leaf inserted there. In reality, there's lots of different kinds of gas exchange chambers that enclose pieces of shoot or needles, entire shoots here, or even entire pieces of tree. But in all those cases, these are all airtight chambers into which we place leaves. And usually there's a source of light that shines into that chamber so that we can measure photosynthesis of the leaves in that chamber. Okay, so we've got this airtight chamber. So let's say we put some tubing. Well, these lines represent tubings and a pump. So we take gas out of the chamber using a pump and pump it through an infrared gas analyzer. And then let's say we then take the gas from that infrared gas analyzer and put it back into that chamber. This is what we call a closed system. So in a closed system, we have a leaf in a chamber. We're pumping air out of the chamber through an infrared gas analyzer and then back into the chamber. So that air is just recirculating through this closed system. So let's think about what the concentration of CO2 measured by this infrared gas analyzer might look like over time. So what I'm going to do is draw what that might look like over time. So let's say we've got time on the x-axis, CO2 on the y-axis, CO2 concentration. So this leaf is in this closed system. So over time, as that leaf photosynthesizes, the CO2 concentration in the chamber will decline over time because that leaf is photosynthesizing and taking up CO2 from the chamber. So over time, the CO2 concentration in the chamber will decline. And that's what that output from the infrared gas analyzer would look like. So let's say we put a different leaf in there that had a higher net photosynthesis rate. What would that look like? So if it had a higher net photosynthesis rate than this initial leaf, we would still see a decline, but the decline would be faster because the photosynthesis rate is higher and that leaf is taking up CO2 faster. So in a closed system, the photosynthesis rate is related to the rate of decline of CO2 concentration in the chamber. One of the advantages of a closed system like this is that it's very simple. It only requires one infrared gas analyzer, and you're just recirculating air. And the calculation of net photosynthesis is simple. Just the slope of that decline of CO2 concentration is the rate of net photosynthesis. There's some problems with this kind of a system, though. So think about CO2 concentration in the chamber here. It's always changing. So CO2 actually affects the rate of net photosynthesis. So you're changing, the leaf is changing the environment in this chamber over time. In addition, that leaf is also transpiring. It's letting off water vapor. So the water vapor concentration in the chamber is elevating over time. So that's one of the big problems with the closed system, is that it's difficult or impossible to keep the conditions, the environmental conditions in the chamber the same. So basically, measures of net photosynthesis have to be taken over a relatively short period of time, so the CO2 concentration and the water vapor conditions in the chamber don't change much during the measurement. To overcome some of those issues, other types of systems have been developed. So it just shows you the time series. So let's think about another type of gas exchange system. So we start out with the chamber again. Now we have a pump that's pushing air from the atmosphere or from some other source through an infrared gas analyzer and into the chamber. And then we have more tubing that leaves the chamber and goes through a second infrared gas analyzer and then vents to the atmosphere. This type of system is called an open system because the system is open to the atmosphere. We're just flowing air through the chamber and out into the atmosphere. What this means is that we can condition the air that goes into the chamber to hold, for instance, CO2 concentration and water vapor concentration constant by doing things to the air, drying it or humidifying it, changing the CO2 concentration so that it'll be at steady state in the chamber and we can hold conditions constant. We have the ability to do that in an open system like this. In an open system like this, these two infrared gas analyzers are called, or the CO2 concentration have different names. So CO2 reference and CO2 sample is what we might call those two infrared, the signals from those two infrared gas analyzers. Let's think about what happens or what those infrared gas analyzers will read for a photosynthesizing leaf. So if we have some CO2 concentration entering the chamber and then some CO2 concentration leaving, this leaf is photosynthesizing, the CO2 concentration of this sample will be lower because the leaf is removing CO2 from air. So the reference CO2 concentration will be higher than the sample CO2 concentration if photosynthesis is occurring. One handy thing about infrared gas analyzers is they can also measure water vapor concentration. So we can use a system like this to not only measure the rate of net photosynthesis, but also the rate of transpiration, the rate of water vapor loss or release from the leaf. So if we're measuring water vapor, and this is water vapor concentration and water vapor concentration here, in that case, if this leaf is transpiring, the water vapor concentration of the air leaving the chamber will be higher than that entering the chamber. So a concept that's useful in an open system is the delta. Delta CO2 is just the CO2 concentration in the sample erga minus the CO2 concentration in the reference erga. And the rate of net photosynthesis is related to delta CO2. At a given flow rate, the higher the net photosynthesis rate of that leaf, the smaller the CO2 sample will be, or the bigger the difference will be, and the bigger the delta will be. So at a given flow rate of air through the chamber, higher photosynthesis rate will result in a larger delta CO2. Okay, so there are different photosynthesis systems that you can buy commercially, and most of them are open systems like this. This is a LCA corporation. These are two types of Lycor photosynthesis systems. And these photosynthesis systems have readouts. So the one that we'll be using in lab has a readout that looks like this. So basically, there's a CO2R, that's CO2 reference, and a CO2 sample. That's the concentration of carbon dioxide in the reference infrared gas analyzer in this one, and then the CO2 concentration in the sample gas analyzer. That's this one. And then it also calculates delta, calculates the difference between these two. So the photosynthesis rate is a function of that delta number. We have the same thing for water vapor. Water vapor in the reference chamber and water vapor in the sample, and the delta H2O. Transpiration rate is this number, and that's calculated from delta H2O. We can also derive stomatal conductance. That's how wide open the stomata are. That's derived from this transpiration measurement. These systems are also handy because they can, for instance, show us graphs of, for instance, photosynthesis over time or stomatal conductance over time, because there's a built-in computer that monitors all of these variables over time and can allow us to monitor changes in, for instance, photosynthesis over time. So in this lab, we're going to be using a gas exchange system to measure net photosynthesis. So I'm going to give you a quick tour of one of these gas exchange systems.